Welcome to the course of plant developmental biology. So, in from previous class we are continuing reverse genetics based approaches. So, if you recall previous lectures, so we have finished identification and isolation of genes for functional study. We have studied already analysis of expression pattern, particularly global analysis or the expression pattern where differential expressed genes has been already identified. In this class we will more focus on the spatial expression pattern analysis. So, this is very important in context with the developmental biology, because a development of any organs or any tissue is a context dependent process. And if you recall our few of the very previous lectures or very one or first or second lecture, we have discussed that cell-cell communication is one major driving factor in case of the development. So, if you want to study function of a particular gene through reverse genetics based approach, the first and very important thing what you have to do, you have to very precisely study what is the expression pattern, particularly is spatial and temporal expression pattern. Spatial means which tissue, which cells or which organs it is expressed, and temporal means at what stage of the development it is started expressing. So, there are several ways to study to analyze expression pattern, particularly spatial and temporal. One way is RNA in situ hybridization. Here you can do in whole mount tissue or tissue section. So, basically in whole mount tissue you can take a full organ or full structure, and then you can probe that with a specific gene specific probe and then identify where is a particular gene expressed. In tissue section what we do, you first fix the section and then make cross sections of the tissues, 8 micron thickness thick tissue or 10 micron thick cross sections, and then you probe that cross section with a gene specific probe. So, this method you can use to identify RNA, messenger RNA, which means that you can identify the transcriptional activity of a gene in a tissue specific manner. Second thing you can study is the protein in situ hybridization, where you can look where a particular protein is present. So, there are other way to study, that is temporal and spatial gene expression pattern, which is promoter reporter assay and promoter trapping. We will discuss one by one. So, first we will take RNA in situ hybridization. So, as I said that here you can do in two way, either you can do whole mount in situ hybridization or you can do on the sections. But what are some steps which you have to follow? So, the first major step of RNA in situ hybridization is tissue preparation. So, you collect the sample depending on what tissue or what organs you are going to study. You can collect root, stem, inflorescence, flowers depending on your interest. Then first important thing is that you have to fix the tissues. This is very important to terminate any ongoing biological reactions or biochemical reactions. There are various fixative available to fix the plant tissue, and then these two processes are common in both whole mount in situ as well as in situ on the cross sections or longitudinal sections. But if you want to do on the sections, then another thing what you have to do, you have to take this tissue and embed in embedding media. This is very important because you want to make a very thin section, which is 8 micron or 10 micron section, and the embedding media more preferably what we are using is a paraffin. So, there is a process of dehydration. This is important, we do through ethanol series to remove the water, and then you infiltrate this with the paraffin, and then once your paraffin blocks are ready, you take these samples, you make a cross section, collect these sections on poly-L lysine coated slides. This is very important because you want your section to stick on the slides and it should not fall off. Then another important thing is that you have to generate RNA probe. As you can see here, this is RNA in situ hybridization, which means that you are detecting messenger RNA for a particular gene and using RNA as a probe. So, if you have to make probe, you will have to make antisense RNA as a probe, because this antisense RNA will go and hybridize with the sense RNA. But as a control, you will also make sense probe to basically check what is the background signal. And how you make these probes, maybe I will describe in detail. There are two ways. One is that you can do PCR based probe synthesis, or you can clone a gene specific fragment in a vector and then use that as a template to generate the probe. Since we are generating RNA probe, the process which involves is in vitro transcription, and here we are using either T7 RNA polymerase or T3 RNA polymerase depending on which promoter is available, and then you can use labeled NTP. So there are two way of doing it. One, the initial way what people used to make probe, they used to make radioactive label probe, and now because of lot of issues with the radioactivity, we are moving more towards the non-radioactive label probe. And in this probe synthesis, what happens, either you take one of the NTP as isotope labeled, or you can use some of the chemical synthesized NTP. For example, this is biotin labeled UTP, or digoxazine labeled UTP. And then this probe, depending on what is the size of your gene, you can use them and you can hydrolyze them. So what you do basically, you take your gene specific fragment, you clone in a cloning vector. The important thing is that your cloning vector should have a T3 and T7 promoter, or you can use sp6 promoter depending on the vector available. And then if your gene specific fragment is in the sense orientation, if you have to generate antisense probe, you will have to use this T3 promoter and T3 RNA polymerase. And then for doing this, what you do, you linearize this vector from here and then use T3 RNA polymerase to transcribe this region as an antisense probe. But while transcribing this region, you are adding UTP, which is tagged with digoxazine, which is called dig UTP. And then if this probe synthesis, if the size of probe is higher, then what you prefer to hydrolyze it and make a probe size of roughly 75 to 100 nucleotide long. This is important because if size of probe is very high, it will not properly penetrate the tissues at the time of hybridization. So for example, this is a gel picture which is showing how to generate the probe. So this is a linearized plasmid. So you have plasmid with your gene specific fragment cloned in it. Then you have done in vitro transcription and you have generated the probes. This is RNA probe, but this probe size is quite high. Then we have hydrolyzed this probe and we have made a small hydrolyzed probe. Now this hydrolyzed probe is ready for hybridization. For hybridization, what we do before going for the hybridization, there are some treatment with the tissues, and one very important treatment there is the proteinase K treatment. This is very important to partially digest the tissue to allow better probe penetration. And then once you have treated your tissues, then you use this RNA probe and do hybridization, usually 16 to 20 hours at 50 to 55 degrees centigrade. Then once your hybridization is finished, it is very important for you to remove unbound probes, otherwise they will also give background signal. So what we do to remove them, one treatment we do which is RNase treatment. Typically we are using here RNase A, which specifically degrades single stranded unbound RNA. So this will remove all unbound RNA from the tissues, and then we do stringent washing to remove even if there is something unhybridized probes. This is very important before going for the detection of the signal. If you do not do this properly, then you are going to have lot of background signals. Then the process goes, once you have already sections, you have already probed with the antisense RNA probe, then you use antibody. So typically we use anti-dig antibody. So you know that our antisense RNA probe is tagged with dig. Now we are using antibody which recognize digoxazine. So this anti-dig antibodies we are using, and then depending on what method or what way you are going to detect it, if you want to do the colorimetric assay, then what we use anti-dig antibodies which has alkaline phosphatase conjugates. But if you want to detect through the fluorescent based detection, then you can use anti-dig antibody with some of the fluorescent tagged protein, for example rhodomine. So basically you have now treated the slides with the antibody, then you have to wash to remove unbound antibodies, then go for the detection. Again the method of detection will depends on what kind of probes, what kind of antibody you have used. So if you have used radioactive probe, then you can detect the signal by autoradiography. If you have used anti-dig antibody which has alkaline phosphatase or other color based enzymes, then you can do the color reaction. But if you have fluorescent tagged antibody, then you can do the fluorescent microscopy to detect the signal. I will give you some of the example. So this is a case of whole mount in situ, and one of the gene which is a rice transcription factor MATS2 was detected or analyzed for its expression pattern using non-radioactive DIG UTP labeled antisense RNA probe, and this was detected by fluorescent microscopy. So just to quickly know that this is the organ where we want to see the expression. This organ is called lodicule. It is a small rice flour organ. You can see here very clearly that this is the organ here, and we want to see that how MATS2 express across the organ. So we have basically taken this entire lodicule and we have done the whole mount in situ hybridization using antisense probe against the MATS2, and then detected using anti-rhodomine labeled antibody. And you can clearly see here that with this in situ method we can tell that expression of MATS2 is asymmetrically distributed. So if this is a lodicule, you can see the tips and the peripheral region of the lodicule have more MATS2 signal as compared to the proximal and basal region of. So this analysis basically not only allowing us to check the expression in a particular organ, but it is also allowing us to check if the transcript is asymmetrically distributed. Similarly, if you take this example, so here basically these were done on the longitudinal sections of the panicle. So this is again rice panicle, and rice panicle is like inflorescence. It has different florets at a different developmental stage, and this upper panel, if you look, they has been labeled with antisense RNA probe for another gene which is called MATS1, but it is a radio labeled probe. So you can clearly see the expression is throughout at early stage, and then later stage it is more restricted to the lemma and palea, and expression in inner wall organs are disappearing. So this is a typical example of radioactive label RNA in situ hybridization on sections. On the other hand, if you look this OSMGH3 is a downstream gene which is regulated by MATS1, and what happens if you see here at this stage, so this is done by non-radioactive labeled antisense RNA probe, and if you look at this stage you can see that very similar to the MATS1, it shows a high level of expression throughout the floral primordia. But in the later stage when the floral organs are organogenesis has already started, you can see the expression in lemma and palea is decreased, but the inner wall organs are having high level of expression. So this way this method is allowing us to study where exactly a particular gene is expressed. So this was example of RNA in situ hybridization, but we all know that RNA in most of the cases the functional molecule is protein, not the RNA. Of course, there are a lot of RNAs which are functioning without translated into a protein, but in general cases or typically RNAs are undergoing the process of translation and they are making the protein. So if you want to detect the protein in a tissue, or if you want to detect the spatial and temporal localization of the proteins, you can perform protein in situ hybridization, which is also called immunohistochemistry. And what you can do, this is a typical example of a whole mount. Here this is quite similar with the RNA in situ hybridization, except the detection method is slightly different. So here also you collect the tissue, then do the fixation, then after fixation you are doing the membrane permealization, and then you can add the antibody. So there are two possibilities. If you want to detect a protein against which you already have antibody, then you can use that antibody. If you do not have antibody against a particular protein of interest, then you can tag the protein. You can tag your protein with some tag. For example, you can add HA tag, MIC tag, or any kind of tag, and then you can use antibody against the tag to detect the protein localization. And then there are two ways. If you have a primary antibody already tagged with the conjugates, if it is already conjugated, you can just use primary antibody. Else you can first detect with the primary antibody, and then you can use a secondary antibody which is against the primary antibody, and then you can visualize the signal. Signal visualization you can do in a different way. You can in fact co-stain with different dye. So for example, if you take this example here, a protein which is PIN1 protein, very important oxygen transporter protein, has been seen for its localization, and you can see the green signal is for PIN protein, and then another molecule or another protein which is basically tagged with the red signal, it is ATPase protein. So basically this is going to mark the membrane protein, and then DAPI, which is blue in color, DAPI basically binds with the DNA, so it is going to stain the nucleus. So these all three together has been used to basically check the localization of PIN protein. So if you look here, so this is maybe enlarged version of it, and you can clearly say that this is the cell boundary, and in the cell boundary the PIN protein is very specifically localized or asymmetrically localized on one side of the cell membrane, but if you look this side of membrane, the protein localization is very small. So this is, I am taking just one example, but there are plenty of examples available where you can detect this protein by protein in situ hybridization or immunohistochemistry. Okay, so next method which is being used to monitor temporal and spatial gene expression is promoter reporter assay, very commonly used. So if you recall one of our previous lectures, so we have discussed that there could be two types of promoter reporter assay. You can either make a transcriptional fusion construct or make a translational fusion construct. What is difference between them? In a transcriptional fusion construct, so you only take the promoter or the upstream sequence of your gene. So if this is your gene, you only take the promoter region, and this construct basically will tell you the transcriptional or promoter activity wherever it is available. And then you use a kind of reporter gene here. Reporter gene, either you can use GUS kind of gene, or you can use GFP, RFP, any kind of reporter gene you can use here. And then if you generate this kind of construct, it is called transcriptional fusion construct, and this construct basically will tell you where is the promoter activity domain. But this alone cannot be used, because in plants most of the time it has been seen that promoters are not very localized. There are some cis regulatory elements which are essential for the promoter activity, and they are even found within the gene, sometime in the introns. So it is always challenging to choose that how much region or how much segment of the DNA to be taken for the promoter activity. So if you take a fragment, let us assume if you are taking 3 kb upstream sequence from the start transcription start site, then you will clone it and you will see a expression pattern. But if you want to validate it, or if you want to be very sure that actually this is the promoter activity, you will have to do this analysis together with the in situ hybridization. So in situ hybridization, just now we have discussed, there you are going and you are not doing anything, you are just detecting the endogenous expression pattern of a gene or of a RNA, and then you are making the transcriptional fusion construct, and when you are using this transcriptional fusion construct, you see where is the expression. Then you can compare if your promoter activity or if your promoter reporter activity is same as the in situ hybridization, then you can say that the promoter element or the promoter region which you have selected, it is complete and it has all the cis regulatory elements which is part of this promoter or which is required by this promoter. In translational fusion construct, what we do, that this is more important because it will tell domains of the expression as well as subcellular protein localization. So, here what we do, we basically take the desired promoter and then we take the gene as well, but we fuse this gene with some of the reporter. So, basically what we are making here, we are mutating the stop codon of the gene of interest, and then in frame we are fusing with some of the reporter, and then what happens that this, and if you use this construct, this is called translational fusion construct. So, this will tell first thing that wherever is the promoter activity it will report, and second thing is that what is the subcellular localization of your protein. So, because your protein is now tagged with the GUS and GFP, so wherever your protein will move, it will report, the reporter will report its presence. But here there is one issue. The issue is that since you are making a fusion protein, there is a possibility that this fusion might inactivate your protein. So, your protein might not be in its the proper conformation, and then if you report its localization, it is always questionable that the localization is because of the artifact or it is the true localization pattern. So, this has to be validation, this you can validate it. How you can do? You can validate your construct by doing the complementation, which means that if you take mutant of this gene of interest where you see some phenotype and then take the translational fusion construct and put back to the mutant. If the phenotype of mutant is complemented by this construct, which means that your fusion protein is functional and you can rely on its localization pattern. Okay. So, I will give you now a few very important examples where transcriptional and translational fusion construct has been successfully used to reveal a very important information. For example, if you look this short root gene. So, short root gene, it is very important for root development in Arabidopsis, and if you do just RNA in situ hybridization or if you generate a transcriptional fusion construct. So, this is endogenous RNA detection by RNA in situ hybridization, and here you have taken short root promoter and fused in here GFP, here GUS. In all this case, if you look the expression domain of short root protein, what you see that this gene is expressed in the vascular tissue. So, this is if you look this is a longitudinal view of the growing Arabidopsis root, this is endodermis. So, if you see this in endodermis you do not see any signal, but the tissue after endodermis like pericycle, xylem, phloem, procambium, they have the activity. This is same you can see here, and this is also you can see when you make when you do in situ hybridization here in the root. So, this tells that the short root promoter is active in the vascular tissue, but when you make a translational fusion where you are using same short root promoter, but now short root protein has been translationally fused with the GFP, what you observe that apart from the expression domain which is in the vascular tissue, you also see that protein is also present in the endodermis, and in endodermis it is getting localized to the nucleus, and this has a lot of importance. Maybe in some of the future classes we will discuss what is the functional significance of this. So, how this is possible? If your promoter is active here, transcription is going on only in the vascular tissue, how come your protein is present in the tissue where transcription is not happening? So, this gives an information that there might be a possibility that your protein is getting synthesized in the vascular tissue, and then from vascular tissue it is migrating to the neighboring cells which is endodermis in this case, and this is only possible if you study both transcriptional as well as translational fusion construct for the gene. Similar kind of study has been done in shoot apical meristem of Arabidopsis thaliana, and here a very important protein which is called ushal. Ushal is important to regulate and to maintain the meristematic activity. You will see in the next class, but here what I wanted to show, if you look the in situ hybridization pattern of the ushal, you will always see that this ushal is expressed somewhere here in this domain, but this domain which is L1 and L2 layer, this is inflorescence meristem, this is floral meristem. In all the cases you can see that L1 and L2 layer, they do not have RNA, which means that transcription of ushal is not happening in L1 and L2 layer. But when you take and when you look the protein localization of ushal, then what you see here, so this is ushal promoter driving GFP fused with the ushal protein. You can see that in L1 and L2 layer, both the layers have the protein. So transcriptionally it is here, RNA is here, and then the protein is also present in the region where transcription, the active transcription for ushal is not happening. So this tells that this protein might be moving from one cell to another cell. Okay, the final method what is being used to study temporal and spatial expression is promoter trapping or enhancer trapping or gene trapping. What is this trapping? This is very important method, and what you can do, there are different types of trapping you can do. This is typical genomic DNA, and then for example you want to identify a gene with a very particular or with a defined expression pattern. How you can do? One way of it, doing enhancer trapping. Here we will focus more on the promoter trapping. I think enhancer trapping we have already discussed in one of the previous class. But what will happen if you have to trap a promoter of your interest? If you want to identify a gene which has, let's assume, expression in leaf, or if it is expressed only in the petals, and you want to identify that kind of gene for the reverse genetic basis study, how can you do? So basically you can generate a construct. In this construct what happened, that you put a reporter gene, but in the reporter gene just promoter less reporter gene, and make a construct in the tDNA and use this tDNA to raise the transgenic plant. So basically what is happening, this tDNA will randomly get inserted in the genome, but if this tDNA got inserted, let's assume in a gene of interest which is expressed in the petal, and if let's assume that if this is the promoter, this is the gene, and here let's assume if your construct got inserted, then now it has got a promoter activity from here of the gene which is like petal express, and then you have a reporter gene here. So basically you are randomly integrating a reporter in the genome and trying to identify a transgenic line where your reporter has been integrated in the downstream of a promoter which is active in a particular tissue, and then you can basically isolate them. Similarly, you can do the gene trapping, you can do the enhancer trapping. In enhancer trapping, what basically we do, we take a minimal promoter and then reporters. So promoter, minimal promoter as well as reporter, and then try to trap the enhancer. Here we are trying to trap the promoter, and you can see some of the example that this has been very successfully used to identify lot of genes whose expressions are in a very restricted manner. For example, this gene is expressed in the leaf, this is expressed in the trichome, then this is expressed in the root apical meristem. Similarly, here if you look, the lateral primordias are coming, lateral root primordias are coming. So basically then since you know the tDNA sequence, you can use some of the molecular biology based technique to map the gene where is the site of integration, then you can identify a gene for functional study. Yeah, here is one very good example where the genes or the lines has been identified which show either petal or stamen, and or some of them are expressed both in petal and stamen. So you can see there has been generated, so large number of gene trap lines has been generated, and if you look these lines, they are having a different expression pattern. Some of them are specifically expressed in the petal or specifically expressed in the stamen. Some of them has been expressed both in petals and stamens. Then you can go map this gene and then identify and take this gene for the further functional study. Okay. So, if I recap what we have discussed today. So, today we have finished the analysis of expression pattern, particularly we have taken what are the ways and to study the spatial and temporal expression pattern of a gene in plants, and particularly these are very important when you are studying development of a particular organ, or if you are identifying some putative regulator of a developmental process, and if you want to study their specific function in the process of development. So, I will stop here. In next class, we will discuss functional characterization of a gene. So, once you have identified a gene, how you will take it for further functional characterization. Thank you very much.